Hello, wonderful audience. Thank you for choosing to spend your time with me. We extend a warm welcome to you as we discuss and analyze SLITRK in this video. SLITRK SLIT and NTRK-like family. Member 1 is a human gene that codes for a transmembrane and signaling protein that is part of the SLITRK gene family, which is responsible for synapse regulation and presynaptic differentiation in the brain. Expression of the gene has been linked to early formation of excitatory synapses through binding with receptor tyrosine phosphatase PTP LAR-RPTP. Various studies over the years have linked mutations in the gene to conditions on the OCD spectrum, Tourette syndrome and trichotillomania. However, the mutations in the genome itself vary greatly between individuals, with most mutations observed being hard to find in repeat studies. Members of the SLITRK family, such as SLITRK, are integral membrane proteins with two N-terminal leucine-rich repeat LR domains similar to those of SLIT proteins. See SLIT, MIM 600 and 3742. Most SLITRKs, but not SLITRK, also have C-terminal regions that share homology with neurotrophin receptors. SLITRK, MIM 191,315. SLITRKs are expressed predominantly in neural tissues and have neurite-modulating activity. Aruga et al. 2003. With the groundwork laid, let's now examine gene and its connections to our previous discussions. The gene for SLITRK is located on chromosome K. The gene is expressed only in the brain of humans. The MNA can differ from alternative splicing and contains domains for the extracellular matrix as well as for the LARs. Mice contain an ortholig of the gene called SLITRK. Brace yourself for a captivating discussion on protein structure as we explore its nuances and implications. SLITRK contains two horseshoe-shaped leucine-rich repeat domains LARs in its extracellular domain which are vital to its function. The LARs have 6 modules each and are connected by a 70-90 amino acid loops. LAR is a more conserved sequence and is present as a dimer while LAR is a monomer and has a more variable sequence. The conserved sequence of LAR contains critical binding pockets and specific charged residues that are important for it to carry out its function of binding to LARs on the N-terminus. Both LR sequences are randomly positioned on the protein and contain variable linker regions. The protein also contains a short intracellular domain, but lacks a tyrosine phosphorylation motif which is present in other SLITRK genes. Without further ado, let's move on to the topic of function. SLITRK is highly expressed in the central nervous system. It plays a critical part in regulating synapse formation between hippocampal neurons and in differentiation of synapses, helping in neuronal outgrowth. It is expressed during embryonic stages and postnatally, but expression decreases over time and is localized to the postsynaptic membrane. Overexpression of SLITRK promotes postsynaptic differentiation for excitatory and inhibitory synapses, but because of the localization, only excitatory synapses are affected. Inhibition of SLITRK only reduces differentiation of excitatory synapses because of this. Let's transition to interaction with LAR-RPTP and uncover its significance. Since they lack tyrosine phosphorylation motifs, SLITRK binds to LAR-RPTP through its LAR region in order to differentiate synapses. The LAR domain's function is not clearly understood yet, but it is hypothesized that it is for dimerization to the cell surface. LAR-RPTP binds to the LAR region through its IG region, with 3 separate binding sites in a binding ratio. IG binds through electrostatic and hydrophobic interactions, IG binds through ionic and hydrogen bonds, and IG binds through hydrogen bonding. The unique properties on the concave surface are what determine which LAR-RPTP binds to it. If the proper LAR-RPTP is not bound to the LAR, then synapse formation cannot occur, but bonding can still occur. Once they are bound properly, the complex is sufficient for synapse differentiation. Point mutations in the LAR region impaired differentiation as well, but not binding. As we transition, let's shed light on Tourette syndrome and its relevance to our ongoing exploration. The The SLITRK gene is not a major risk gene for the majority of individuals with Tourette syndrome (TS), according to a 2009 review, although its study can help contribute to our understanding of TS. Rare variants in SLITRK may lead to TS, and mutations in non-coding regions of SLITRK may also play a part, but further research needs to be done before any conclusions can be drawn. In 2005, medical researchers observed a de novo translocation in a patient with TS which broke the patient's chromosome near the SLITRK genome. In screening of additional patients, the authors observed a frameshift mutation in SLITRK in a patient with TS and the same REMKNA target variant called VAR and VOX target of miR24-1 in two patients with TS. These variants were not found in several thousand controls supporting an association of the variants with TS. A subsequent examination of the region of the SLITRK gene found the mutation in none of 82 patients with Tourette syndrome. The authors concluded that tests to detect variants in the gene probably would have little diagnostic utility. An experiment in the effects of a microdeletion in chromosome was done in a fetus. The mother had passed the microdeletion to the child and both did not have Tourette's or any other OCD symptoms, showing that it may not be a direct cause of Tourette's. Further attempts to replicate the study were done in multiple studies. In a Japanese study, next-gen sequencing was used to screen 92 TS patients and 361 healthy controls. None of TS patients were found to have mutations at either variant or any new mutations in the gene. In a European study, it was found that the two original variations were not found in any of the 222 trios that were studied. However, tests were also done on SNPs in the groups and 3 were found to have variations. 2 of the 3 variations were found to be associated with the formation of Tourette syndrome. In a different study of 381 Caucasians with some form of OCD with 356 non-OCD control patients, 3 genetic changes were found after genetic screening. Of the 3, 2 were identified only once each and the third was found in 4 OCD patients but also in a non-OCD patient. The non-OCD patient did have compulsive nail biting, but these studies show that a genetic link between SLITRK and patients with TS may exist. They are more complex in nature than previously understood. Let's zoom in on trichotillomania and understand its implications. The SLITRK gene has also been implicated in a small percentage of cases of trichotillomania, an impulse disorder in which individuals compulsively pull their own hair. In one of the previously mentioned studies, the mother of the child who had a de novo translocation had trichotillomania. This would suggest that there could be a genetic link between SLITRK and trichotillomania as well. A study was done in which 44 families with individuals who had trichotillomania had their SLITRK gene sequenced. 2 new non-synonymous mutations were discovered about 9 base pairs apart from each other, in an area separate from the one where the Tourette mutations were found. These results were compared to control and none had the mutation, suggesting that these mutations, while rare, were associated with trichotillomania. I'm here to provide you with the best information, so let me know what else you'd like to learn about.